Hi everyone, I'm Romane Poinsot from MS Nutrition and I'm pleased to introduce MS Food, a shiny web app designed by us. We are a team of 3 PhD and 1 PhD student based in Marseille. Our daily activities are doing diet modeling and statistics in the field of nutrition and public health. We are not real designer or application developers, but we use the potential of Shiny and R to develop practical tools like MS Food. So MS Food was developed following emergence of Nutri-Score. It's a front-of-pack label which assesses nutritional quality of processed food, and the score is calculated from the content of 4 unfavorable components in red and 3 favorable components in green. So it's been designed for agribusiness professionals, and MS Food helps them compute, compare and improve the Nutri-Score of their food products. A first feature consists in computing the Nutri-Score for a single product. Users need to tick the contents of the 7 components, and then the app automatically gives the Nutri-Score. The control widgets were made by us, starting from the inline radio button provided by Shiny. And Nutri-Score can also be computed for several products by importing a database. A file check was programmed to ensure that all information is available. And a second feature is data visualization. On this graph designed with ggplot2, you can see the repartition of products according to the Nutri-Score, and colors mark out the 5 different classes of the Nutri-Score. And thanks to gggraph package, user can click on one point and see the products corresponding to the points and in more details in the table below. On the following graph you will see, it's a box plot for a quantitative variable such as a nutrient or other variable that the user may have added, for example, the price. And on this box plot, you can see that carbohydrates significantly increase according to the Nutri-Score class. All the charts and tables are exportable in a PowerPoint file thanks to the Office R package. Users just need to click on the plus sign next to chart on table to add as many slides as wanted to the final report. And this last feature consists in improve the Nutri-Score of a product. User just need to choose the product to improve, and then enter the target class, for example, B, if the product is already C, and choose its calculation mode. To quickly assess the feasible solution, user can choose the automatic mode. A solution is a new combination of components that reach the target class. So user just need to tick the nutrient that you would like to be modified with the checkbox button and click on validate. Then the app automatically gives the solutions to what she asked and plot the 50 closest solution to the original products, and the relative distance between the original values and the new solution are plotted on y-axis. And a detail of the modifications are plotted in a modal window. The graph were designed with ggplot2 package and show the original value and the new content range needed for reaching the new value. So the free version of MS Food is available, and also a commercial version is available on our website. So please contact us if you have more questions or if you are interested in getting a demo. Thank you.
Good morning. So I'll be talking about a project where our analysis of the winter pressures in the healthcare system in the UK, and hopefully I will show you how we used R and Shiny to make our analysis a lot faster, to make it more reproducible, and how it also allowed us to visualize our results in a much more impactful way than we could have done before. So for a little bit of background, winter is quite a challenging time for the healthcare system in the UK. And that is because there is a lot more demand for healthcare than what the system can cope with. And one of the results is what I'm showing on the graph here, is that patients are waiting a lot longer in hospitals than they should. And this has been getting progressively worse over the last few years, and every winter it gets quite a lot of attention in the media. So during winter, the NHS releases public data on the situation in hospitals, and that comes from around 150 acute hospitals in the UK. And one of the things we do at the Health Foundation is we try to provide policymakers with better evidence of the quality of care in the UK. So we take this data every week and we turn it into information on the current situation in the healthcare system. We look at trends over time. And we also look at variation between local healthcare systems to see whether there are any difference between how the system is coping. So you can imagine that if you want to do this every week by hand, this is a lot of work. So what we did last year is we developed a workflow in R that would do this analysis and visualization for us. And this year, we would also like to embed an interactive Shiny app into our reporting on the winter pressures. And throughout this whole process, we're doing this to also promote the amazing NHSR community who are trying to get people to use R in the healthcare system. So one of the challenges that we had in this analysis was that a lot of things change every year, and that's the data quality, the layout of the spreadsheets, which indicators are in the data, and even local healthcare systems themselves change and evolve all the time. Another challenge we had was that missing data was not coded consistently. So we think this data is submitted manually, and this means that sometimes missing data is coded as missing, and sometimes it's coded as a 0. So we then had to go in and figure out what was actually missing and what was truly a 0. And this is, of course, highly context-specific and really depends on the indicator you're looking at. So we spend a lot of time figuring out a data cleaning approach that would not throw away any real information and still make our results comparable over time. So this year we would, as I mentioned, so in the end our workflow ended up being quite simple really, and we relied on some of the workhorse packages of R. But it allowed us to turn this data over within a matter of minutes, and it also made the visualizations of the results much more impactful. So I'm sure just a quick look at this map will tell you that bed occupancy within hospitals in England was highly variable last winter, and that the situation got progressively more serious as winter went on. So this year we want to move to Shiny, and we think this is a step up in how we report on the winter pressures, because we are able to incorporate some historical data on the last few years, so to compare how the situation looks this year compared to last year's. And you can also visualize indicators side by side, because often there can be a relationship between them. And in the second tab, we want to then aggregate this data within local healthcare systems to look for variation between them. And we're still trying to figure out how granular we really want to go with this and what it will be most useful for people who'll be using the Shiny app. So I'll just like to finish with some lessons that we learned. And that was really that even a very simple analysis like this involves lots and lots of small decisions that impact what your results look like and how they should be interpreted. So we decided to do something slightly different this year, and we published 2 reports. One of them was a blog where we looked at the results of the analysis, and that was done by my colleague Lucinda Allen and published on the Health Foundation website. And the other report, we then also wrote a Medium blog where we take you through our analysis step by step and also explain the reasoning behind what we did. So that can be found on Medium. And the code for both the workflow and the Shiny app are available on GitHub. If you want to find out more, get in touch or come and chat to me. Thank you.
Welcome to my talk. I'm Thomas Petzoldt from University Dresden. I work for an environmental faculty, and then I hope my slide will come in. The topic is about antibiotic resistances, and you see the picture on the bottom that my normal background is more aquatic ecology. So how does this come together? It was done with some co-authors from Finland and from Australia. You all know that antibiotics are extremely important, and they are so important that Sir Alexander Fleming got knighted. Yes, you know, Great Britain is a very traditional country, and they value efforts, but Fleming already discovered that antibiotics are not without problems, and nowadays there is really a big rumor in the press and in the media that there is a crisis, yeah? Like in the talk before, crisis. So many citizens die each year because no antibiotic is available. So this is dealt, of course, by the clinical context. There are many people and organizations, international organizations, who deal with the clinical field, and that's a surprise, use still Excel and manual inspection, and that's not feasible for us as environmental sciences. We need automatic tools. Here is how antibiotic resistance testing can be done. We take an agar plate, put in bacteria and different antibiotics, and if you see an inhibition zone, then it's working, and if you say no inhibition zone, then it's not working, and this diameter can be measured. This is then done many, many times, so we get hundreds and thousands of isolates. And in addition, there's also, of course, biochemical and molecular biological identification, quality control, and so on. And what we get in the end is a multimodal distribution. So the clinical community is almost only interested in subdividing the so-called wild type from the rest, because this determines if we can apply an antibiotic and in which dose. But we as environmental scientists are also interested in separating substrains. And here you see data from the environment. The bottom is from an airport. You see an increase of antibiotics, especially in the intermediate, so slightly resistant subpopulations, and you see a shift. The most simple method to deal with this is kernel density estimation. On top you see the standard method, but as we have a boundary, that's the disk size of the antibiotic test is 6 millimeters. We need a special method, and that's package EV mix. I forgot to mention it here, but I'm very strict in citing work of others. And this is another algorithm that's used by the antibiotic resistance scene. They have an Excel table to classify the ECOF values, the epidemiological cutoff value, and it was 2 hours to implement it as a Shiny app. So, then we wanted to do more and separate also the other peaks. Here we first use a heuristic peak hunting algorithm, and then maximum likelihood fitting the whole distribution, consisting of an exponential maybe, and then some normal distributions. This is the result, and we can estimate an ECOF. We are not allowed to call it this way because that's the official term. We called it MXCOF, cutoff based on mixture values, and we get estimates for the other subpopulations. This is the comparison with the reference method, the Excel method, only for the ECOF, and I've never seen such an agreement with my work and work from others without calibration, and it's not calibrated at all, yeah? But of course there's just quantiles. So it's automatic, scriptable, yeah, and gets more information than the original one. This is just a placeholder for the results that are appeared since 22nd of June when I had to submit it. So we are in the meantime able to see this shift in nature, in the field here, a comparison between different places in a sewer system in a city. And if you have more questions or want to have a look at the slides, which will be then there here, I forgot to mention BBMLE was used for maximum likelihood fitting. Thank you.
Good morning, everyone. So I will now give a brief overview on how I use R to perform Mendelian randomization studies. And yeah, I will start just thanking Paul Brennan, Matthias Johansson, Shama Virani, and all others working on Mendelian randomization in the genetic epidemiology group of the International Agency for Research on Cancer. So Mendelian randomization is a specific case of instrumental variable method when the instrument is actually a genetic variant or can be a set of genetic variants. So when their genetic variants, they are strongly associated with a risk factor, let's say body size, we can use them to study the association between body size and an outcome like risk of cancer. And because germline genetic variants are randomly assigned at conception, they do not depend on exposure like lifestyle exposure or environmental exposure that can bias other observational epidemiological studies. So what I use to perform MR studies are genome-wide association study results, and which collect genetic information for a sample of individuals and produce summary data for the association between the genetic variants. It can be up to 10 million genetic variants with phenotype, in this case could be body size. And here we can see that we need information on some identifier in the first columns, and then which is the reference and effect allele, and then the estimate of the association with the phenotype. Then you can see how the data look like. But then if I want to answer the question, does obesity cause ovarian cancer? What I would do is a two-sample Mendelian randomization study. And then I will use a first GWAS for BMI to define the instruments, so the genetic variant that are associated with BMI, and a second GWAS for ovarian cancer. In simplifying the analysis step, the first step will be some data preparation using packages that are not specific to genetic data. And then the main package for two-sample Mendelian randomization will be two-sample MR. And I will first use it to harmonize the two data sets, so ensuring that reference and effect allele are the same for the two data sets for all the genetic variants. And then we'll using methods there specific to Mendelian randomization for getting MR estimates and complementary analysis. And then we sometimes use also some meta-analysis packages to perform additional analysis. And then for the visualization plus the data, different packages and different methods can be used. And then now I'm going to show some of the routine result that we can get. For instance, the first plot is directed from the two-sample MR, which requires itself the ggplot2 package. And here we can see there is a scatter plot of the effect of each genetic variant on BMI and on ovarian cancer. So we have more than 700 genetic variants that are associated with BMI there. And then the line there interpolated the data are showing the association between body mass index and ovarian cancer according to different estimators. But it's clear to see that there is an association between BMI and cancer if you're just maybe plotting in a forest plot, like in the second plot, using ggplot2 the result for the different estimates. And another analysis that is routinely done is leave one out analysis, where the estimation is repeated, leaving one one genetic variant out a time. And then here is just a histogram of the result for each leave one out result. So I think that so I want to conclude that when we are using genetic data to study a relationship between a risk factor and an outcome, we can easily done with Mendelian randomization in R. And this is avoiding some of the limitation of other observational studies. And large GWAS data are getting more and more available, but large sample size are still needed. And I wanted to stress that actually assumptions need to be carefully assessed. So if you have any comment on these, feel free to contact me or to just come and talk to me at the coffee break or at the poster session. Thanks for your attention.
Hello, everybody. Sorry for my bear voice. I'm not a bear, I promise. Toulouse greeted me with some light virus. But anyways, my name is Volha Tryputsen, and I'm a statistician at Janssen R&D, which is a pharmaceutical part of Johnson & Johnson. And today I want to tell you about one of the Shiny apps I developed for the group of oncologists working with in vivo data, and the app is in vivo LDA. So at Janssen, we discover and develop drugs, and it takes a long time, on average 10 years or so, for a single drug to be approved. And usually the time is split between preclinical research and clinical research, which is clinical trials. And a part of my responsibility at Janssen is to provide statistical support for the group of biologists who conduct animal studies, and by those, they're testing different compounds in preclinical phase, and also test different treatment or different drugs that are participating in ongoing clinical trials. And what the team really does is they conduct animal studies in mice, where animals are implanted with some tumor type, and then the tumor grows, and the animals are being split into different groups, and different groups are being assigned different treatments. So very simple setup. And then the animals are being followed through the time, and several things are being measured. Number one thing is, of course, tumor volume, so we do care about the tumor size. And then also a body weight of an animal, and some survival data as well. And on the left-hand side of two plots, you can see that each line is an animal, and each panel is a treatment. And then what the scientists are really faced with is look at all the data and draw some conclusions about efficacy, safety, and survival, and choose what treatment works best. And one way to do it, of course, is to take the longitudinal data and compare mean profiles between different treatment groups. So what scientists had at hand is the software that they used in-house, which is GraphPad Prism, where they would conduct their longitudinal data analysis. And they quickly understand that software has limitations. When they have missing data, the software cannot handle the analysis. And that's, of course, is a problem because there are a lot of missing data in oncology studies. And then once we dig a little deeper, we understood that the method itself wasn't the best, and there was no model diagnostics. And altogether, the whole analysis was time-consuming because biologists were doing it themselves, and they're not statisticians. And then the question of reproducibility and traceability is also comes in place. And it was this Monday morning when several of those studies landed in my inbox, once I inquired further, like, how many studies do you do a month? They're like, oh, 25. And I'm like, okay, so that means I need to analyze 23 more, and they're all of a similar design, so the solution for me was very evident. I need to create a Shiny app. And what we can offer as a statistician, of course, we can deal with missing data, no problem with that. And we're gonna offer a more powerful method for longitudinal data analysis for sure. We're gonna create model diagnostics because those are important for checking your model assumptions. And we're gonna wrap all this in a custom-made software, and it's gonna be efficient, traceable, reproducible, and here you go. We developed the in vivo LDA Shiny app as a collaboration with the group of oncologists. So now the Shiny app is used globally for in vivo oncology team, and it's used for all preclinical programs in oncology preclinical space. So instead of, I already mentioned that we do have a better, more powerful approach with linear mixed effect modeling and other ways of looking at longitudinal data. We also offer diagnostics that are very important to assess assumptions. Plus, we do have nice interactive and animated graphics within the app. We can't show it in the slide talk, but they're there. And those are great for exploratory data analysis. And then this is what I care as a statistician, but what biologists really care is to get their stats report official with everything they need within less than 5 minutes. So that was a full success, and even though we had some resistance with Shiny apps at the beginning, now the whole team is so up to Shiny apps, and they requested several more apps for different analysis that they're doing, and this year we're working on another 3 apps. And just before I left to the conference, I was actually notified that this app received one of the Janssen Innovative Leadership Award in the company, which is obviously a great success. And these are some colleagues of mine who helped me to bring the app to the state it is now. If you have any questions, please find me and ask those. Thank you.
So good morning. I'm going to present a Shiny web application that we have developed to fit some of the most usual spatial and spatiotemporal disease mapping models. This is a joint work with Tomás Goicoa and Lolo Ugarte from the Public University of Navarra. So, spatial and spatiotemporal analysis of count data are crucial in epidemiology. You are going to see in the next slide. I'm going too fast. Are crucial in epidemiology to provide accurate estimates of mortality or incidence risks, are to unveil the underlying spatial and spatiotemporal patterns. However, fitting these models could not be easy for non-expert users. So here, I am going to present an interactive web application named SSTCDR, which has been designed for two main purposes: to perform descriptive analysis in space and time of mortality or incidence risks and rates, and to fit an extensive range of fairly complex spatial and spatiotemporal models for aerial data commonly used in disease mapping. This application has been built with Shiny and relies on the well-founded integrated nested Laplace approximation technique for Bayesian inference through the R-INLA package. And there are the two main references. The first one is for the application, the second one is for the INLA approximation strategy. So, users can access directly the application from the web browser without installing any software in their computers, since all the computations are made in a remote server. In addition, a desktop version is also available to run the application locally if needed. Every new user must register for the first time in the application, which is structured in four main parts: data input, descriptive analysis, model specification, and results. In addition, an additional tab with a user guide and some set of tutorials are also available in the application. So, first, the data and its associated cartography must be uploaded by the user, which are automatically previewed on the screen. And several formats for both data and cartography files are supported by the application. In a second step, in a second tab, the variables of interest must be selected by the user, and classical estimation measures such as standardized mortality ratios, crude ratios, or standardized rates are computed. And some descriptive graphs of these variables could be easily generated by the application. Since the great variability of these measures, the great variability of these measures makes necessary the use of statistical models to smooth risks using information from both spatial and temporal neighbors. And many different models have been applied in this application using the R-INLA package. So, in the results tab, summary measures are provided for the posterior distribution of model hyperparameters, relative mortality or incidence risks and rates, and spatial, temporal, or spatiotemporal patterns. We can obtain different graphs and tables that are easily exported by the user. If the interest lies in analyzing the evolution of the geographical distribution of a disease, maps with the posterior means of relative risks and posterior exceedance probabilities of being greater than a threshold value can be generated. In addition, we can also compute or plot the area-specific temporal risk evolutions of the area of interest. In this plot, we have shown the posterior mean estimates and 95% credible interbands, and the color of the bands are associated with the posterior exceedance probabilities. So, I think that the key advantage of this application in comparison with other software commonly used in disease mapping is that it provides an easy-to-use interface that facilitates the use of these fairly complex models without installing any software in user's computer. Some of the future development of the application will be the integration of simple feature objects as cartography files and to include interactive data visualization graphs. We want also to implement other new spatiotemporal model proposals that have been proposed or in the literature. So I just want to conclude saying that this application has been developed together with the spatial statistic groups of the Public University of Navarra, and an email account is available for any bug reports and support for the users. So thank you very much for your time.
My name is Goknur. I am from Melbourne, Australia. I work at the Medical Research Institute. I do postdoctoral research there. I use statistical modeling to explore genomic data sets. So with my colleague, Alexandra, we developed a tool to enable our collaborators to visualize their biological pathways related to their disease of interest. So what was our motivation is for them to create this application, so it will enable them to explore their data more. And because in early research, focus was only one gene, but with the emerge of microarray RNA-seq technologies, now we can focus on more than one genes and relationship between those genes. Like in this example, there's BCL2 gene, but we are also interested in the interaction of BCL2 with other genes. And even further research showed us it is important to construct the pathways of human, and each node here, each blue node here in this slide is a set of genes, not just one gene, and the interaction between those are shown with these blue lines as well. So it is a massive network, but we would like our collaborators to dig into this by using a set of genes they are interested in, and we explore with Pathway Visualizer those set of genes. And it is an interactive, reproducible, and shareable tool for them to share with their collaborators as well. So how does that work is the biologists can input a set of genes as a text file, or they can input a count matrix they obtained from RNA-seq experiment or any other next-gen experiments. Or they can analyze their data using a package. We use Lemma here, and then they can input a fit object. And based on the gene list they provided, in the next step, we can, this program identifies the significant pathways obtained from the general database like Genontology or Reactome, KEK database, whatever they're interested in. And in the next step, we get a pathway hierarchy from Genontology database, and we create matrix of edges and nodes. Then we will be able to create networks between those pathways, which are significant and only includes the set of genes user provided. And we used, to construct this network, we used Wiz network package. Wiz network package allows us to build a network like you can see at the left-hand side. So this only includes the significant gene ontologies that includes the inputted list of genes. So it is also interactive, and you can click on each single dot here represents a pathway. You can zoom in and you can see the details of this pathway as well. The blue ones are the pathways. They are connected to the purple ones, which are selected as an interest. So what else you can do with it is you can, beside this network, you can obtain a table of whatever pathways are you selected interactively, and you can click on those IDs of these pathways, and it will take you to the website, and also it will produce a list of genes included in this pathway. One more feature is not limited to these features, but just to show you an example, you can select from a dialog box which pathways you would like to show in your plot, or you can just select one gene and it will highlight all the pathways including those particular gene, so you can see the connection between them. And yeah, I'd like to thank to my supervisor Gordon Smythe and Melissa Davis for her feedback, and thanks for listening. Any questions? We can talk after the talk. Thank you very much.